Sterilization. Sterilization is making a substance free from all microorganisms both in vegetative and the sporing state. Spore. This is the reproductive structure that is adapted for dispersal and surviving for extended period of time in unfavorable conditions. Spores form part of the life cycle of many bacteria, plants, algae, fungi and some protozoa. So here we can see the terms which are used in the sterilization process. The first one is the disinfection. The destruction or the removal of all pathogenic organisms capable of giving rise to the infection. So disinfection, they does not affect the spore state organism. And next term we can see is the antisepsis. The term is used to indicate the prevention of infection, usually by inhibiting the growth of bacteria in wounds or tissues. This is done by the antiseptics. Chemicals or the disinfectants, which can be safely applied on skin or the mucous membrane to prevent the infection by inhibiting the growth of bacteria. Bactericidal agents or the germicides, those which are able to kill the bacteria. Bacteriostatic agent only prevent the multiplication of the bacteria, but they remain alive. Cleaning, important preparatory step before the sterilization or the infection by removing the soil and other dirt. Decontamination, the process of rendering an article or area free of contaminants including the microbial, chemical, radioactive and other hazards. Methods or the agents of the sterilization. The first agent is the physical method, dry heat sterilization, moist heat sterilization and the sterilization by radiation. Chemical method like gaseous sterilizations and the sterilization by the disinfectant. Mechanical method like they pass through the bacteria-proof filter. The first method among the physical method is the dry heat sterilization. The instrument we use is oven. They are specially designed instrument, electrically heated and thermostatically controlled, exposed at 160 degree Fahrenheit for 160 degree Celsius for 1 hour. Advantages like it is suitable method for sterilization of substance destroyed by moisture. They do not require water and there is not much pressure buildup within the oven, unlike an autoclave, making them safer to work with. They are suitable to be used in the laboratory environment. They are much smaller than the autoclave but can still be effective. Disadvantage: as they use the dry heat instead of moist heat, some organisms like prions may not be killed by them every time. Moist heat sterilization. The instrument we can use is the autoclave. Heating process in the autoclave, they are saturated steam under the pressure is allowed to penetrate through the material for 15 minutes and the temperature is 121 degrees Celsius. Advantages: microorganisms are killed most efficiently in the lesser time due to the high pressurized saturated steam. The disadvantage is unsuitable for the material, notwithstanding the temperature of 115 degrees Celsius or more during the heating. Sterilization by radiation. Two techniques involved. One is the alteration of chemicals lead to form new compound in the cells, destroying the microorganism itself. Vital structures like the nuclear proteins are destroyed, killing the microorganism. Example: cobalt-60. They are used for the gamma ray sterilization process. Here we can look the two types of radiation. One is ionizing radiation and another one is non-ionizing radiation. In the ionizing radiation, we use the gamma rays especially. X-rays, they are used for sterilizing plastics and syringes, while gamma rays, they are generally obtained from the radioisotopes like cobalt-60 during the disintegration of unstable atom. They kill the microorganism by isolating atoms of the essential substance of cell present in them. The advantage of this ionizing radiation is there is no significant rise in the temperature, the continuous process due to the short exposure time. While speaking about the disadvantage, they may lead to color change, solubility of preparation leading to the decomposition of certain materials. Next we can see the non-ionizing radiation. Infrared rays. They are used for rapid mass sterilization of the pre-packed items such as syringe, catheter. Like UV rays, they are used for disinfecting the enclosed areas such as entryways, operation theatres and labs. Testing the efficiency of radiation. Radiation control indicator disc. We can use this instrument for the amount of radiation we are using. and to check the quality of the radiation. Special paper sticker, which is yellow in color, is sticked on the article. After proper sterilization, this color changes to red. Chemical methods of sterilization. The chemical agents we use and the action of chemical agent, like the protein coagulation, disruption of a cell membrane resulting in the exposure, damage, or the loss of contents. Removal of sulfur group essential for the normal functioning of enzyme and the substrate competition. Commonly used chemicals are alcohol, ethyl alcohol, isopropyl alcohol. They must be used at the concentration of 60 to 90%. Isopropyl alcohol used in the disinfection of the clinical thermometer. Methyl alcohol is effective against the fungal spore, treating the cabinets and the incubators. Methyl alcohol is also toxic and inflammable. Aldehyde, formaldehyde, having the bactericidal, sporicidal, and have the lethal effect on the virus. They are used to preserve the anatomical specimen, destroying the anthrax spores on the hair and wool. Glutaldehyde, they are the effective against tubercle, bacilli, fungi, and virus. They are less toxic and irritate to eye, skin. They are used to treat the anesthetic, rubber, face mask, plastic, and the metal instruments and the polythene tubing. Dyes. There are two types of dyes we can use. One is the aniline dye and the acridine dye. They are bacteriostatic in the high dilution but are the low bactericidal activity. Aniline dye is more active against the gram-positive bacteria and the gram-negative organism. Some important dyes like proflavin, acrylflavin, euflavin, aminocrine. These impair the DNA complexes of the microorganism and thus kill or destroy the reproductive capacity of the cell. Halogens. Iodine. They are used as a skin disinfectant having the active bactericidal activity and moderate the action on spores. Chlorine. They are used to disinfect the water supplies, swimming pools and food and the dairy industries. Along with the hypochlorites like bactericidal, they also act on the viruses. Phenols. These are obtained from the distillation of the coal tar between 170 to 270 degrees Celsius. The lethal effects are the capacity to cause cell membrane damage, releasing the cell contents and causing the lysis. Low concentration will precipitate the protein. Gases. Types of gases used for sterilization is ethylene oxide, formaldehyde, and the beta propylene. Ethylene oxide, the gas used is safe and non-inflammable. Nowadays, ethylene oxide most widely used gaseous sterilization agent in the medical science. Action is due to the alkylating the amino, carboxyl, hydroxyl, and the sulfur group in the protein synthesis, also on the DNA and RNA. The advantage is it has penetration power, quite useful for sterilizing surgical instruments such as catheter, needles, plastic, and disposables. Disadvantage is very slow sterilization process, very costly equipment. Formaldehyde gas. This is widely used, employed for the fumigation of the OT and other room. This is produced by adding 150 gram of potassium permanganate to 280 ml of formalin for every 1000 cubic feet of room volume after closing the window and other outlet. After fumigation, the doctor should be sealed and left unopened for 48 hours. Beta propylactone, product of ketone and the formaldehyde with a boiling point of 163 degrees Celsius, having a rapid bactericidal activity but carcinogenic, capable of killing all microorganisms, but it is very active against viruses. Disinfection, decontamination, removal of microorganism contaminating an object. Preservation, preventing the method of microbes caused spoilage of susceptible product, pharmaceuticals and foods. Sanitation, removal of microbes that pose a threat to the public health, food industry and water conditioning. Sanitizer, an agent, usually a detergent that reduces the number of bacteria to a safe level. Mechanical method, the solution to be sterilized is passed through the deep filter, screen filter, which includes a particular filter, microbial filter, final filter. The pharmaceutical solutions are sterilized by this method. The microorganisms are physically removed by the absorption on the filter medium or by the mechanism. Sterilization test must be done. Thank you.